You're listening to CoachingCues podcast, where experts all around the world answer your most burning questions surrounding the wide topic of strength training. Every week we tackle the what, the why, and the how of one specific topic in just 15 minutes. Straight to the point, no fluff. So, without further ado, let's get straight into this week's episode.
Hello guys and girls, my name is Henning Wackerhage. I'm Professor for Exercise Biology at the Technical University of Munich. And for this podcast, I tried to discuss the stimuli and the senses that initiate skeletal muscle hypertrophy following resistance exercise. So, trying to discuss the stimuli and the senses that initiate skeletal muscle hypertrophy after resistance exercise in 15 minutes is a little bit like trying to discuss world history in half an hour, because you can never be totally complete with this topic. If you have a great interest in this, so come to Munich and study our master's course in sport and exercise science, because there we really discuss it in all detail. Okay, so what is the problem? So I assume most of you, and I know many people in Uvascular, go to the gym, lift heavy weights, and their muscles grow. The question is, okay, how does this work? So what do we know and what do we don't know? So what we know is that whenever you lift heavy weights, mTOR or the mTOR complex 1, mTORC 1 gets activated, and that then stimulates the ribosome, which is a machine in your muscles that produces new protein. And this was discovered by Keith Barr and Karen Esse, because they found out that sort of this mTORC1, at least proteins connected to it, they became activated when muscles did resistance exercise. What we also know is it's not only mTORC1 that gets activated and then switches on the ribosome, and that increases protein synthesis. And because muscle is 70% protein, so if protein synthesis goes up and exceeds protein breakdown, your muscles will hypertrophy. But in addition to that, from gene expression studies where people use gene expression microarrays, so where you can see whether the transcription of a gene is increased or decreased, what we know from these studies is that also when you go to the gym, you lift heavy weights, your muscle is switching on and off an awful lot of genes. So it's not only that mTORC1 gets activated and that this activates the protein synthesis by the ribosome, but at the same time, something is switching on and off genes in the muscles that you have just trained. So a big question in the field is, what is actually the stimulus associated with resistance exercise that activates mTORC1 and that causes all these changes in gene expression that together then change your muscles and cause muscle hypertrophy? And in relation to that, 3 stimuli are mainly discussed today. The big one is, of course, mechanical stimuli, because when you go to the gym and lift a heavy weight, you generate a mechanical force. That force pulls on your muscle, and of course this could be a key stimulus. However, then people did things like venous blood flow restriction and resistance exercise, and found you can actually exercise with quite low weights and still get a hypertrophy. And so people thought, well, this points us in the direction of some metabolites, so metabolic stress, which might also be a stimuli for muscle hypertrophy. And then the classical one is you go to the gym, you ask people, so why does resistance exercise stimulate muscle hypertrophy? Some people will tell you, well, it's a damage you need to cause, because the damage then triggers muscle hypertrophy. So who's right? Is it the people who say it's mechanical stimuli? Is it the people who say it's metabolites? Or is it the people who say it's exercise-induced mechanical damage? Which is the stimulus that actually initiates muscle hypertrophy? And if there's a stimulus, there needs to be a sensor. Yeah, like for everything visual, you need the eye to perceive this. Equally, if there's a mechanical stimulus that initiates muscle hypertrophy, you need a mechanosensor in your muscles or elsewhere that senses that there was a load on the muscle, and that then activates mTOR, and mTOR then switches on protein synthesis. Or if it also regulates gene expression, then there needs to be a link from the mechanical stimulus to a sensor to a change in gene expression. So in this relatively short podcast, I will discuss the evidence for mechanical sensors. So when we look at the literature on this, the important studies early on have been done by Goldberg. And he came up with a model of Synergist Stipulation. So a synergist is a muscle who has a similar function like another muscle. And what he did is he took one of these two muscles away, and as a consequence of that, the other muscle, the synergist, when you take one muscle away, then the other muscle has to produce more force, because it has to cover for the muscle that you've just removed. And what he found is that this was enough to cause muscle hypertrophy. So this is very different than traditional resistance training, because if you do synergist stipulation, then the overloaded muscle is overloaded permanently, probably not with 80% or 70% or 60% of the one repetition maximum, but much lower. And it is overloaded not for 10 repetitions or so, but for maybe 2 weeks. So this is very different than going to the gym and lifting heavy weight. And so people take this as evidence that such a mechanical overload, that the mechanical overload is actually the stimulus. But hang on a minute, is it really? Because if you mechanically overload a muscle, that muscle has to work harder, and so it will change its energy metabolism because it needs to resynthesize more ATP to be able to exert a higher force. Yeah. So it's not only just a mechanical load that you put onto that muscle, but in addition to that, in its response, the muscle also needs to alter its metabolism. And then maybe also you cause damage. So you can't really conclude from that experiment it's a mechanical load which is causing the hypertrophy in a synergist ablation rodent model. The second evidence for mechanical load as something that regulates muscle mass is when you break your leg and the doctor puts your leg in a cast. As a consequence of that, you will take away the mechanical load and your muscle atrophies, suggesting that even if you have that basic load, which is far away from the one repetition maximum, the basic load of just standing and walking around, that is sufficient to generate a certain amount of normal muscle mass. Because if you take this normal loading pattern without going to the gym, this very normal all-day loading pattern, if you take that away, your muscle atrophy, suggesting that this normal loading pattern already causes some sort of muscle growth so that your muscle looks normal and not atrophied like when the muscle is in a cast. And so again, mechanical load seems to be the obvious stimulus here, which you take away and then you get the atrophy, and if you put it back on, you get the normal muscle size again. But again, if you take away the normal loading, you also change the energy metabolism of that muscle. And I don't know whether anything happens to damage or so, but it is not just a mechanical load that you manipulate. And finally, of course, it's resistance training in a gym. Picture Arnold Schwarzenegger or some bodybuilder, male or female, lifting heavy weights. You load your muscles mechanically. But every time you lift a heavy weight, you also need to resynthesize far more ATP because you have a high ATP usage, so you affect metabolism, and there's many other things going on. And again, you can't say it's mechanical load which is causing the hypertrophy. So to summarize, there are several experiments where people conclude this is evidence that mechanical load is a stimulus that initiates muscle hypertrophy after resistance training. But actually, when you look at these experiments, it's not only mechanical load that has been changed in these experiments, but many other factors. And so the conclusion is, we can't really 100% say it's mechanical load. In my view, it's very likely that mechanical load is a key stimulus, and I'm sure you all agree. But I don't think we have absolute proof in experiment that shows with no doubt that mechanical load is a stimulus that initiates muscle hypertrophy in response to resistance exercise. So this brings me to the next question. So is there actually any evidence that if we change the mechanical environment of a cell, that actually the cell responds to it? Can cells respond to mechanical load? Let me give you one example. So in one experiment, cancer researchers have been growing the same MDA-MB231 breast cancer cells in a soft matrix and in a hard matrix. So it's a mesh, and it is stiff on the right hand side and it is soft like jelly on the left hand side. And what they found is these cancer cells, the same amount of cancer cells, the cancer was growing much better when it was implanted into the stiff matrix, and so suggesting that the cell can sense a mechanical environment, whether it's soft or stiff, and that it then responds to that by more growth or less growth. Okay, then there was an experiment by Engler in the Journal of Cell Science in 2004. So he put myoblasts onto a super jelly surface, really, really soft, then about the hardness of a muscle, and then really stiff like concrete. And what he found is that these myoblasts differentiated best into myotubes, which is a sort of like a muscle fiber, when the stiffness of the surface was about as hard as a muscle fiber. So this suggests that even when muscles develop, they really depend on the stiffness probably of the environment of the so-called extracellular matrix, because if it's too stiff, they don't differentiate well into muscle, and if it's too soft, they neither do that. Okay, so in summary, there's some evidence that cells can sense mechanical load and change their behavior accordingly. So let's take a closer look to muscle. So if we take our microscope or electron microscope and really magnify a muscle, what we will see is a sarcomere. So a sarcomere is a myosin and the actin which interact to generate force, because a myosin head, they pull on the actin, and each myosin head produces a force of 6 piconewtons. And so because there's millions and millions and millions of myosin head, each one producing a force of 6 piconewtons, this all adds up, and at the end you're lifting 100 kilograms. So, okay, now the question is, in the system, in the sarcomere, do we know that there are some proteins which start signal transduction in response to loading? That the protein either gets pulled on, gets stretched, and then it starts to phosphorylate another protein, or when there's a force on the protein. So how do we have to imagine this? And there's actually evidence for mechanosensitive proteins that might initiate signaling. And that might be located at the Z-disc or in the so-called costameres. So from the Z-disc and from the sarcomere, there are protein complexes that transmit the forces generated by the sarcomeres to the extracellular matrix, which is surrounding muscle fibers. And so this is a dystroglycan complex, and there are also the so-called focal adhesion complexes. And in these focal adhesion complexes, there's a focal adhesion kinase. And in these, on the Z-disc and in these costameres, so these protein complexes that connect the sarcomere to the outside extracellular matrix and help to transmit forces to the outside, there are some force-sensitive proteins. So I don't have a chance to discuss them all. Focal adhesion kinase is one. Titin is force sensitive and responds to it. I want to focus on one of these complexes because I think this is a really good candidate. So, and this is the filament C BAC3 complex. So filament C is a protein and BAC3 is a protein. So filament C looks like a V, like victory V. And actually here in Munich, there are some crazy physicists, and they are able to pull with a highly sophisticated method on single molecules. And what they found is that the filament C, which is on the Z disk and which connects the actin to the Z disk, and that the filament C is opened up at 20 piconewtons. So now remember that one myosin head produces a force of 6 piconewtons. So super small force, but because you have millions and millions and millions of them, you really can lift those 100 kilograms. And so you can imagine if 4 myosin heads generate a force of 24 piconewtons, and if that is pulling on one filament C, which is V-shaped, then the filament C opens up, and so this might initiate some signaling. And actually what we know from the research is that Troy Hornberger and his team found that filament C also gets phosphorylated after really high intensity exercise. So the filament C now is connected to BAC3, and BAC3 has a so-called WW domain. So let's assume, okay, you lift that heavy weight, the filament C opens up, then something happens which we don't know really, but sort of the BAC3 then somehow becomes activated, and we also know BAC3 becomes phosphorylated after high intensity contractions. And so BAC3 has this WW domain. So W stands for tryptophan, don't ask me why, a W stands for tryptophan, but this is how it is. And so this is a little bit like Lego bricks. You need these two tryptophans that the BAC3 can dock onto other proteins which have a so-called PPXY motif. Yeah, so imagine two Lego bricks. So the two tryptophans, they're like these little things sticking out of the Lego brick, which you need to stick it to another Lego brick. And so this is a WW domain interacting to a PPXY domain. And so interestingly, TSC1, which is an inhibitor of mTORC1, has such a PPXY domain. And so Sebastian Gehlert and colleagues, they did a biopsy study, and they got some initial evidence so that when you do resistance training, then presumably that filament C and that BAC3, they become activated due to the force generated. And then the BAC3 via its WW domain pulls somehow away the TSC1 with the PPXY domain from the mTORC1. So now you can imagine you pull the inhibitor away from the mTORC1, and then mTORC1 gets active because the inhibitor is gone due to the signaling I've just described. And so the mTORC1 would then activate the ribosome and elevate protein synthesis. So this would be the model. But BAC3 also has an effect on so-called Hippo proteins. And the super interesting thing to me is that these Hippo proteins regulate the LUT1 leucine transporter. And this is really interesting. And we know from other studies that this leucine transporter, LUT1, gets more expressed after resistance exercise. So this suggests it's not only that this filament BAC3 TSC1 complex activates mTORC and protein synthesis, but at the same time filament C and BAC3 after loading activate these Hippo proteins, and they then produce more LUT1. And so as a result of that, the muscle you've just trained in the gym has more LUT1, it can take up more leucine, and the leucine will then activate mTORC1 and further increase protein synthesis. So this is what I wanted to cover today. So without showing slides, I can imagine that this is quite complex, but the take-home message is, yes, cells can respond to mechanical signals such as the stiffness of the environment or such as a load on a muscle. The specific example is the filament C BAG3 complex. The filament C is like a V, and if you pull on it, it opens up at a force of 20 piconewtons. Now, as one myosin head produces a force of 6 piconewtons, you can imagine if 4 myosin heads generate a force of 24 piconewtons and pull on the filament C, which opens up at 20 piconewtons, the filament C will open up. We know that when you exert a heavy load, when you do a high intensity contraction, that filament C and BAG3 get phosphorylated. So we assume this might activate these guys. And then the BAG3 has two functions. It pulls an inhibitor away from mTORC1. So this is like in your car, you take the foot off the brake. So presumably mTORC1 gets more activated. It switches on the ribosome and the muscle produces more protein. And if you have more protein in the muscle, that muscle will hypertrophy. At the same time, BAG3 also pulls away an inhibitor of Hippo signaling. So this Hippo signaling becomes active. And the active Hippo signaling then produces an amino acid transporter termed LAT1. And as a consequence of that, more leucine will go into the muscle, and leucine activates mTORC. So you have a second mechanism that will activate mTORC1, and so mTORC1 then switches on the ribosome further, and you get even more protein synthesis. Right, so my big question is, did that work as a podcast? Do you have an idea how it might work? I hope that you got the idea that there are three main potential signals or stimuli that initiate hypertrophy after resistance exercise. It's mechanical load, it's metabolic stress, and it is exercise-induced muscle damage. Today I focused on mechanical load, and I tried to explain the filament C, BAG3, TSC1, mTORC, and the pathway that activates protein synthesis, and also the link via Hippo proteins and the leucine transporter LAT1 that might get more leucine into the muscle and further activate mTORC. So thank you very much. If you have any questions, send me an email. Thanks a lot. Bye.
Thank you for listening to CoachingCues podcast. If you would like your question to be answered by an expert, please head to coachingcues.org/ask. See you next week.